biology. So we have three groups here. Group 2 and Group 3 is on Tuesday. Group 2 is from 8 to 10, and Group 3 is from 10 to 12. And we have another group, which is Group 1, which is on Thursday, from 2 to 4. For today, which is a week, I want to introduce you with some of the basic terms of developmental biology, then followed by developmental genetics, experimental embryology, and followed by cell-cell communications. And we're going to talk about the model organism, which includes the zebrafish, mouse, C. elegans, Drosophila melanogaster, and probably chip, which is the Gallus. And on week 6, we're going to talk about the fertilizations, which is the beginning of new organism. And week 7, we might have midterm, or we might not have. But I just put it there so that we may have some option there. All right. Then we have central nervous system with the CNS on week 8, followed by neurocrystal, the CNS and axonal specifications, and lastly, which is plant development. And we're going to have the experiment, right? Experiment 1, 2. We'll probably have another 2 because we need to comply with the COO, which I will show you in the course plan later. And we'll probably have online presentation, which I put there as optional, right? And this is our cost plan. Okay, sorry, the cost outline actually. So if you look at the COO, we have 2 COO in this course, which is the distinguish the concept on the various aspect of developmental biology, and also demonstrate the basic techniques in developmental biology. And this one, for this one, that will be the practical. The practical, I'm planning to give you 4 practical. Actually, you should have 4 practical, which is the lab report that consists of 40%. So one lab report will be the 10%, which is total up to 40, which comply with the COO2, whereby the COO1 is for assignment, written assignment, and also the final assessment. So altogether, COO will have 60% and COO2 will be 40%, right? And these are the units of learning units. We have 4 learning units here with a total of 80 hours, okay? And for online asynchronous teaching, we're going to have the 28 hours, right? So we have 14 weeks, 10 2, so altogether with the 28. And these are the latest references. I mainly use Woolfolk, okay, and also Gilbert, and also Essential Developmental Biology by Jonathan Slack. Jonathan Slack is my professor at UC of Bath, where I did my PhD. And of course, we have developmental biology from Nadia, and also for principle of development genetics, which is by Sally Moody, right? These are the references that you may refer to for this course, right? Right. I think that's all for the brief introductions. Then I'm going to talk about, we need to go to the lecture now. But before I start, do you have any question?
So far, no, doctor.
Okay, good. No questions. So, before I start, let's look at this way. So, what do you really understand about developmental biology? Do you have any idea of what developmental biology is all about? Any idea? What is developmental biology?
Growth.
Growth, okay. I have 40 students and only one that respond to me. How about the rest?
To study the process of development in
Okay.
living organisms. Yeah.
Okay, that's very good. What else?
And understand the developmental abnormalities so we can know how to cure it.
Okay. Any more? Or you prefer just to keep silent?
Investigate the size and the shape of the living things.
To get what?
Investigate the size or shape.
To investigate the shape and size of the living things. Okay, what else?
Reproduction.
Okay, where we talk about reproduction, of course. Any more?
The study of molecular life.
The molecular?
The study of molecular life.
Okay. Any more? Okay, right. Most of the answer actually is in front of you, like cell signaling. You know, we're going to talk about this. The embryo molecular experiments. You know, the mutagenesis, and many more. All right. All right. Thank you for those who responded. I noticed on that. Right now, let's talk about the principle in human biology. Right. So what did the subject come from? Where did this come from? Actually, this is about in just before 1980. Not much is about knowns about this subject, but in just the short of 30 years ago. So the biology has become one of the most exciting area in biology research, and these advances actually come from three main concepts, which is the experimental embryology, developmental genetics, and molecular biology. So this is the combinations of these three main concept that comes up with the new term of the biology. Okay, and if you look at this illustrations, do you agree that at the very beginning of life, you can see the lizard, the tortoise, the pigeon, the pig, and also humans, they are very similar in terms of the shape.
Yeah.
Yeah.
Yeah. So that's mean you look like a lizard. Yeah. At some point, isn't it? Of life.
Yeah, we all have tails.
Do you agree with that? At some
Yes,
point.
yes.
Yes, at some point.
At some point. But when people say you look like a pig, you don't get angry. Yeah? Because at some point of life, at the beginning of life, yes, we look quite similar. But along the process, then we developed. We have different phenotype. We look different. Yes?
Yeah.
Yeah. Yeah. So don't get angry when people talk about that. When people, you know, don't get angry about that. So learn the value from science is very important as well. Right? Now, in the developmental genetics, and actually it's been flourished. Okay, in the late 1970s, we missed the mass genetic screens has been carried out on the fruit flies or the melanogaster. Okay, Drosophila melanogaster, and thousands of mutations were examined. So it's very interesting to decipher the function of the genes, and based on the expressions of this gene, then they term the gene with different name. For instance, if you look at this ABCDGFHI, You can see that in C here, they call peanut. Yeah, they actually look like this is actually the skeletal, okay, signaling that you can see that this the why the term peanut, probably the expression here, if you look at this, is actually look like what? Like a peanut, isn't it?
Yeah, kind
Shape.
of.
The shape. This is how scientists give the name of the gene. It's based on the expressions like short or capo or and I don't know what's the house is that, but scientists can be very crazy. This is how they name it. Yeah. So identifications of the high proportions of genes that control the development, not just in Drosophila, but in all animals. This is the dream of all scientists, that we want to decipher the function of the gene. Which gene control which part? We want to understand the function, not the network of the gene. So if you look at the Drosophila here, you can see we have the normal one and we have the mutant type. So you compare the mutant Drosophila to the normal one, you can see they are very much different in terms of the phenotype, isn't it? Yeah. Yeah. Phenotype is an appearance. Okay. We have genotype, we have the phenotype. So I hope you can get back to your general genetic so that you know what is genotypes is all about, what is genotype is all about. Okay. Phenotype and genotype. And if you look at the molecular biology, then it started with the discovery of the 3D DNA in 1953, and it's become the practical science of gene manipulation in 1970. So if you look at this, it's this embryo. Can you see the embryo here?
Yes.
Okay, if you look at the full embryo here, you can see the purple color. Can you?
Yes.
Yeah.
Okay, the purple color actually indicate the locations where the gene is expressed. So if you look at that, the genes are very much expressed in the somite. The somite is a muscle for the fish. That's mean, what does it mean is that because the gene is strongly expressed in the muscle, in the somite. So what can you deduce from this expression? What can you tell from this expression of the gene? Why you cannot see that in the head, isn't it? Not many blue, purple color in the head, isn't it?
Yeah.
Yeah, but they are very much expressed in the, very strongly expressed in the somite, isn't it?
Yep.
So what can it tell to us? What can we deduce from this expression pattern?
It keeps expanding.
What is that?
It keeps expanding.
Okay, any more? Why the gene only expressed in the muscle but not in other part of the body? So what can we tell from this? No idea? Okay, you must remember that the gene only expressed when it is needed. Not all genes will express from the moment you be formed until you die. No. Gene only express in the spatial and temporal way. They only express when they are needed. When they are not needed, they are not expressed. So from this picture, you can see that the gene is very important for or responsible for the development of the somite. And that's why they are expressed strongly there. Do you understand?
Yeah.
Yes.
Yes.
And
Yes.
this gene, they are not needed in the anterior part, which is at the head part. That's why you cannot see any blue color there. Yes?
Yes.
And this is what how we study gene expression. Gene, if they are strongly expressed in that specific domain or location, that means this gene is very, very important in that part. They have a very important role in that part. If not, they will not be there. Do you agree?
Yes, agree.
Yeah, logic or not?
Yeah, it makes sense.
Make sense kan? Kalau dia tak perlu, kenapa dia perlu ada di sana? Kalau dia di sana, maksudnya dia mesti adalah dia punya peranan yang penting di sana, betul tak?
Betul.
So, ini logik lah. Betul tak? So, kita kena pandai, kita kena tahu kenapa jin. Jin itu sangat bijak. Dia hanya berada di sesuatu tempat dan express dia punya itu kerana dia ada peranan yang penting di sana. Pada sesuatu masa. Kalau akan pada sesuatu tempat. Kalau tidak, dia tidak akan express. Jin tidak kepoci. They use a very economical way to run the system in our body, in life. Okay, dan lepas tu kalau kita tengok this is cross section di depan ni, right, ini expression gene yang lain lah. So kita pun tengok, right, ini bukan gene yang sama ya. Ya ini dengan ini tak sama. Ya ini kalau kita cross section kita pun nampak banyak blue color di sini. Ini bermaksud bahawa gene ini yang ini gene lain lah untuk yang dekat somat ini. Dan juga yang cross section ni adalah dua gene yang berbeza. That's why kalau kita tengok ekspresinya memang berbeza, betul tak?
Ya.
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 Kalau kita tengok cross section memang, okay, dia banyak berada pada mata, pada brain juga. So maksudnya yang gene yang di sini untuk yang ini dia mempunyai peranan yang penting untuk the eye, maybe the eye developments or maybe the brain developments. And also here we have this, the cloning, the cloning devices here that you can see. You can clone into the vector, and from the vector you can do the transformations. So this is advanced into the cloning, the nucleic acid hybridization, and also the DNA sequencing. And here is the DNA sequencing. Okay, kita boleh tengok sequence. This is on the gel, agar gel, and then we can sequence. You can see the ATCG bagaimana jujukan genetik itu boleh dikeluarkan. Okay. Right, untuk central positions in biology. So the development biology unite the discipline of the microbiology, cell biology, genetics, and also the morphology. So the first process of development create a certain symbol of morphology, which then serve as a basic in which the further rounds of signaling and responses can occur, and eventually to create a more complex morphology. If you look at this, this is the activations of the transcription factor during the gastrulation, and this is the normal development. Okay, the genes featured of MT1, MT2, and RTX, they are actually expressed. Why? Because they are needed. Right? They are needed, and at the end, they slowly to the organizer. Then they establish the three different layers. Okay, after the later differentiations, we have the mesoderm, we have the ectoderm, and we also have the endoderm, which is not shown here. So in the mesoderm, we have the notochord with the somat, we have the kidney, and also the blood vessel, whereby in the ectoderm, which is the most outermost layer, we have the neuro and epidermis, so which at the end, it becomes the skin of the organism. Okay. And the impact to the society, what is the impact to the society is that, you can have the in vitro fertilizations. Yeah. Have you ever heard of this? In vitro fertilization?
Yeah.
Okay, this is it's a good news for those who are incapable to conceive. So they can use IVF to assist them. And we also have artificial insemination by the donor, and also the egg donations, cryopreservation, and embryo transfer. These are the technologies that we can apply based on the study of biology. Okay, and the science of the technology studies the effect of the environment agents such as the chemical, okay, the viral infection and radiations on the embryos, and understanding the chromosome basics of the human birth defect. Contohnya, right? Kita ada Down syndrome. Down syndrome, how many pairs of the genes there? How many pairs of the chromosome yang dia ada? Or counterfeital. Masih ingat tak?
23?
Okay. Kita ada 23 pairs kan?
Yes.
Yeah.
Yes.
Kalau counterfeital macam mana? Kalau apa tu syndrome Down macam mana? So you sendiri pergi ingat dan masih ingat tak general genetics? Macam mana? Kalau untuk syndrome Down tu
Extra gene.
dia ada extra kan? Extra gene ke extra chromosome?
Sorry, extra chromosome.
Yeah, commercial.
Kalau counterfeital? Masih ingat tak counterfeital? Pernah dengar tak counterfeital syndrome? So I hope you still remember lah ya. So future impact, which is understanding the development of mechanism, will assist the pharma industry in designing the drugs against the cancer and degenerative medicine. So now you actually can screen your own genome, and then you can actually design the drug based on your own genome, what you call the pharmacogenomics. You can tailor-make the drugs for yourself in the near future, and even now. For certain people, they can actually tailor-make their own drugs, because not all drugs are effective for everyone. Yeah, so they need to tailor-make them. And then the in vivo functions of many signal transduction pathway can be visualized in organisms such as the xenophis or zebrafish or Drosophila or Xenophagus elegans. These are the animal models that we use to understand the signal transduction. Okay. So the possibility of using our understanding of growth and regenerative process for therapy, contohnya ya, for regenerative medicine. So we can regenerate certain organs by using the contohnya organ culture. Yeah, the organ culture you can, for example, if you lose your ear because of accident, then you can culture the whole ear and then you implant it back. And this is what you call the regenerative medicine. Okay, or the extensions of the existing parental screening to encompass a whole variety of the single gene disorder. You can check whether you have any possible gene disorder or not, and also possible applications of the biology to productions of the human tissue or the organ for the transplantation, such as the tissue engineering or embryonic stem cell. Yeah, pernah dengar tak stem cell, embryonic stem cell? And tissue. Yeah, contohnya you can culture the stem cell, and after that you can treat certain diseases or sickness using the stem cell. For example, our Dato' Lee Chong Wei, because he hurt his tendon is mistaken, then they use the stem cell technique so that it can heal much faster, so that the stem cell can grow into the tendon cell and then replace the injured one. Yeah, this has been done very widely now if you have money. Yeah. If you afford to do that, and also to produce a pharmaceutical in the milk of sheep, and also the vaccine in the egg. Okay, and there are many more actually. If you read more, there are a lot of quality of products that's been produced because of the understanding in developmental biology. Okay. And the common features of developments is they happen mostly in the course of embryonic and developments as the fertilized egg develops into the complete organism. So the concept in development can be loosely classified into 4 groups, which is the regional classification, cell differentiations, morphogenesis, and also the growth. These are the basic 4 domains or concepts that we study in developmental biology. Another one is the genomics equivalence or the cloning animals. So in any animals, the sperm and eggs and their precursor are known as germline. Yeah. And other than that, they are called somatic cells, right?
Yes.
Okay. And the germline cells are to return the full complements of the genes, otherwise reproduction will not be possible. That's why you see these genetic material are very well kept in the nucleus of the sperm and also in the eggs. Yeah, but can we use the somatic cell to generate a whole new organism? Is that possible?
No.
Can, how many say can? How many say no? Let's see. Let type in the chat box. You just type can and cannot. Let me see. I want to see how many. All right, very good. There is many say no. Let me see how many say can. Majority say cannot. Okay, Adeline Fatini, you say can. Can you give me one example? Adeline? See you can. See you also say can. Okay, see you. Can you give me one example also?
Blood cells. Oh, the blood cells.
Blood cell.
Yeah.
How?
Uh,
I mean from the somatic cell. Somatic cell, yeah, blood cell is a somatic cell. How can this cell create a whole new organism? Is that possible?
Actually, I'm not sure. Sure.
Okay, it's okay. Okay, never mind. Let me show you something. Let me draw you something, right? Right. Do you know Dolly the sheep?
Yes.
Yes.
I believe you heard of this story, right? Dolly the sheep,
Yes.
right?
Yep.
Do you know how Dolly was created?
Cloning.
Cloning. Do you know
Yes.
how the cloning was done?
Mm.
No idea, right?
Yeah.
Okay, right. Let me show you something. Can you see my screen?
Yes. Yes.
Okay. This is a somatic cells. In these cells, okay, you have a nucleus. Okay, this is a somatic cell, and this is the nucleus. Okay, this is the end, right? Can you see that?
Yes.
Okay, now this is an egg. Inside the eggs, they also have the nucleus, isn't it? This is also the nucleus, isn't it? Okay, this is an egg or ovum. So how Dolly was created is that they used a technology by removing the nucleus inside the ovum, okay, and then this is what we call a nucleated egg, okay, a nucleated egg, and then they extract this nucleus, okay, right, this nucleus, and by using the electrophoresis, electric force, they push this inside here, use the electric power, electric pulse, right? So this is a nucleus from a somatic cell, right? You get me?
Yes.
Yes.
Then this one, they culture it, put in a plate, they culture it, and this cultured ovum or transferred into the surrogate mother. And this is how Dolly is made. Is that clear?
Yes.
Yes.
Now, is it possible?
Yes.
Yeah.
Yeah, the nucleus from a somatic cell can create a whole new organism. All right. Are you clear now?
Yes.
Yes.
All right. But Dolly, they die very soon. Do you know why? Any idea? The preserved Dolly. Dolly actually was made in Roslin institutions in Edinburgh, in one of the royal institutions in Edinburgh, and they preserve it and put the body of the Dolly was put in the Natural Museum of History in Edinburgh, and I visited the place before. So it's quite cute, but too bad it's died too soon. Do you know why? Any idea?
Short lifespan
Why short lifespan?
because cloning.
Because of the disease.
Why?
Is it
Dolly
because
is-
it have, sorry,
So, yeah, go ahead.
is it because it have lower immune system? I
May
mean,
I know?
since the disease,
May I know who is speaking now?
Ian, sorry, yeah,
Very
and
good, Ian. Very good. Very, very good. Yes, that's the answer. Yeah, because if you look at normal organism, well done. If you normal organism, okay, it's created by sperm, fusion of sperm and also ovum, isn't it? Yeah, and the sperm and ovum, they have the nucleus inside there, and then they have the immunity, all the immunity information inside. So when they become the normal zygote, then they have the immunity. But for Dolly, okay, for Dolly, no. Okay, totally do not have this immunity. They have very, very weak immunity, and that's why it is easily being infected by all the microbes and they die. Yeah, because they don't have, because inside here, if this is the somatic nucleus, and they don't have this immunity information. Is that clear? Logic?
Yes.
Yeah, very good, Ian. That's a very good answer. Yeah, well done. I believe you read a lot on that. Yeah,
Thank you.
well done. Right. So yeah, basically this is how a genomic prevalence and how cloning is done. Yeah, I believe Dr. Chong will teach you in depth on this because you are taking animal biotechnology, isn't it? With Dr. Chong. Yeah. So it's good. So you can he'll teach you. I don't want any developing not overlap information. So let him teach you more on that. Okay, I focus more on the developments of the animals. Okay. Right, but there are some exceptions to the genomic equivalence. For example, the antibody-forming genes of the B lymphocyte and the T cell receptor genes of the T lymphocyte are known to undergo rearrangement of the genetic level. Okay, not every cell is going to do that, but for certain cells, there are some exceptions. And certain nematodes, although not the C. elegans, shed the chromosome from the small some cells line during development. And also the polyploidy, where the whole organism set is double or quadruplet, can occur in some mammalian tissues, such as in our liver. And the polygyny, where the DNA replications occur repeatedly without the chromosome divisions, which leads to the giant chromosome, occur in some tissue in the Drosophila. Okay, so these are the exceptions in the genomics equivalence, right? And gametogenesis is the sexual reproductions of means that life, well, involved in unions of the male and female gametes to form the fertilized eggs. So we all know that, yeah, each gamete derived from the cell contribute to the haploid chromosome set, so that the zygote is also diploid. Okay, so during the embryonic development, germ cell undergo a period of multiplications and migrate from sites where their formations to the gonads. And after the germ cells arrive, they become fully integrated, and in the post-embryonic life under the gamete formations, gametogenesis. And in gametogenesis, we can be divided then into the oogenesis and spermatogenesis. Yeah, so if you look at male and female oogenesis and spermatogenesis, they're slightly different, whereby from primary spermatocyte, you can successfully get the mature of four spermatozoa. But for oogonia, at the end, they only have one mature ovum, compared to four mature healthy spermatozoa at the end from a primary spermatocyte. So another three of the cells will be degenerated. So they're left with one ovum. Okay, so that's the difference between the oogenesis and spermatogenesis. And I believe you have probably have learned this, yeah, in the past. Okay, have you come across with this? Have you learned this before?
Yes.
Yes, we have.
Yes.
Okay, so we'll just skip this, not to talk about that. So early development. Okay, and we look at the early developments. The animals is small and spherical and polarized, and they have the upper hemisphere which carry out the polar bodies. Okay, which is called an animal polar body, which is the animal hemisphere, and also the lower hemisphere, where we call the vegetal hemisphere. And they go through the cleavage as well, from one cell into two cell, and from two cell to four cell. Some of the organisms, they go through symmetrical cleavage. Some animals, they go through asymmetrical cleavage. It depends. Yeah. And also, the early cell divisions are called cleavage with the productions of the blastomeal, okay, blastomeal or blastula or blastocele. Okay, this is a blastocyst. So many species of embryo they remain inactive during the cleavage phase, and the protein synthesis is directed by the maternal mRNA. So at the very early cleavage or the very early activities of the embryo, most of the proteins are actually from the maternal contribution. Okay, until the zygotic transmissions is occur, then the embryo itself will produce its own protein. But before that, it all depends on the maternal genome, the maternal contribution, yeah, of the mRNA. So you must bear in mind that at the very early life or the very early cleavage, yeah, the proteins to allow all the informations are from the maternal mRNA, but not from the zygote itself. Okay, so this is the stage whereby the maternal effect, where the embryonic cleavage that depend on entirely on the genotypes of the mother. Okay, so so during the cleavage phase, a cavity usually form in the center of the ball of the cell due to the uptake of the water and become known as the blastocyst. This is what we call the blastocyst. Okay, and the blastura is a cell that being bound by the catecholamines and will usually have a system of tight junction forming a seal between the external environment and the internal environment of the blastocyst. They have been tightly joined together. Okay, and gastrulation then convert the simple ball, a sheet of the shell, into the three-layer structure known as the gastrula. Right, so this the cell movement heavily occur here, establish three different layers, which is the ectoderm, endoderm, and mesoderm, right, as see here. And of course, in the middle, we have the acantron cavity. All right, so far okay, so far ada soalan, any question? Boleh faham?
Yes.
Okay, boleh ya? Okay, kalau boleh, now we tengok paksi, the axis of and the symmetry. So if animal and vegetables are at the top or the bottom, then the equatorial plane is horizontal plane dividing the egg into the animal and vegetal hemisphere. So you see, this is on the top of this is the animal pool. We have the vegetal pool here, and this is the ventral, this is dorsal, dorsal ventral, and you have the left and right. So if you divide them into these equatorial planes, you have the medial planes here. Okay. And this is a frontal. Okay. So if you look at an animal here, okay, this is the embryo. So the head part is what we call the anterior. The tail part is called posterior. Okay. And the upper part and the lower part, we call this is the top, this is the bottom. So this is dorsal ventral. And we have the sinusolysis and the situs inversus. Okay, this is how the orientations of this internal structure. Okay, and the frontal lines is often, but not necessarily the plans of the second cleavage. Okay, and then the head end is the anterior, as I said just now, and the tail end is what we call the anterior posterior. So the head to tail axis is called anterior posterior. Okay, whereby the top bottom axis called dorsal ventral, and the left to right axis called medial lateral. Okay, daripada kiri ke kanan, we call it medial lateral. So you must be very, very clear with the axis punya term. So that bila saya cakap dorsal ventral or anterior posterior, you know what I refer to. Okay, but in human anatomy, because we stand upright on two legs, so the term anterior posterior is normally synonymous to the dorsal ventral. Because we stand upright, right? So from head or bottom to from the top to bottom, okay, is also dorsal ventral, but we also is also referred to the anterior posterior, right? So the term of proximal and distal are also usually used in relation to the appendage. right? The proximal meaning the near to the body, and also for distal, that means this is for the away from the body. Okay. So untuk exit ni dia you clear tak? Bagaimana kita nak term dia?
Yes, doctor. Doctor, I have a issue. I don't think the slide is moving.
Huh?
Yeah, we are staying at the first page of axis and symmetry.
Ayo,
Yeah.
how come? Ini mas punya wifi teruk tu. Not moving. Memang. Sekejap sekejap. It's not moving also sekarang.
Not yet.
How about now?
Yes, can, doctor.
Yes, can.
Okay, good, thank you. So are you clear now with what we're going to term the anterior posterior, the dorsal ventral, and medial lateral? Okay. Anterior, posterior, dorsal ventral. Okay. Boleh faham
Boleh.
Yes, boleh.
So ini pun sama ada. Okay. Ini pun sama. So this is a repetitive actually. So the morphogenic, okay, the morphogenic process is a cell shape change, and movements are the fundamental to the early development. So cell will move. So you can see here we have the junction here where that's tie the cell together, link the cell together tightly so that it will not be like dispersed everywhere. Okay. And we have also the microfilament bundle contractions here, like the laminar proteins and the focal contract here, and with the substrate. And with the cells, we have matrix, which is the mesenchyme, and also we have the apical contractions, whereby you can see the apical microfilament bundle, whereby they change, changing the cell shape into disc. Okay, and for the cell adhesions, the vertebral epithelial cells are bound together by tight junctions and adherence junctions, and also the desmosome. So the adherence as a major adhesions component, and if the cell of different adhesion system, they are mixed, will sort out into the separate zone. So you can see here, okay. So you can see here, yeah. There are different color, yeah. So you can see there are different system. But all these cells, they are tight. They are actually joined together by the tight junction and adherent junction and desmosome. If they are not tied together, they're linked together. So what will happen to the cell? Dia berkecai berai, betul tak?
Yeah.
Yeah. So it's very important for this cell they hold it together so they make a complete body. Semua sel kita kena ada macam ni, betul tak?
Yes.
Yeah.
Yes.
Right. So this is the classical classifications of the morphogenic process. You can see the condensation, how the cell they condense become like this, and cavitation, macam mana dia cavitate, and then the involute, okay, involution, invagination, and also the epithelium to the mesenchyme. Okay, these are the classifications. And the mesenchyme to the epithelia, for the epiboly, macam mana dia boleh jadi bulat, and then the convergent extension, bagaimana dia daripada square kepada convergent, lebih memanjang, elongated, then juga branching of the morphogenesis, how they branch out. Okay, these are the classifications of the morphogenic process. And this is the growth and death lah. You can see, I believe you have seen this before. So this is like a cell cycle, okay, whereby you can see the G1 is a resting phase, then go to the growth, then we go to the DNA synthesis, go to growth, and then go to the cell divisions, which are involved in the mitosis, whereby you have the metaphase, anaphase, telophase, and there are 4 phases there. Right? So you can see the cell divisions from 1 cell into 2 cell into 4 cell. This one is like a symmetrical division, but not all cells there are symmetrical divisions. Like this one in B, there are asymmetrical divisions. They can see you have this one, the big one is a small one here. All right. Or you also have the exponential growth, whereby you have the 1 cell become 2 cell, 2 cell become 4, 4 become 8, 16, 20, 64. 128, and also the stem cell. All right. I think this is our last slide. Yes, this is our last slide for today. So do you have any question?
No question.
Wow, so lihai ah. No question ah. My explanation is too good. Everybody understand ah. Betul ke?
Excuse me, doctor.
Yeah.
I have a question. I don't really understand the part of axis and symmetry because in the slide here it says in human anatomy, they said that the slide says that if the anterior and posterior are represented in human anatomy, it is similar to the dorsoventral part. So I don't really understand that sentence itself.
Okay, don't worry. We'll come back here.
The next slide. Yes, the third sentence.
In human anatomy.
Yes.
Okay, you see, we stand upright, right?
Yes.
Okay, our head is anterior, right?
Yeah.
So our foot is posterior, right?
Yes, that's correct.
So
and then I don't understand this. What does it mean by synonymous with dorsoventral?
Dorsoventral adalah atas ke bawah.
Okay.
Top to bottom. So our head is top also kan?
Yes.
Our kaki bottom kan?
Okay.
So because in all organisms only humans stood upright, right?
Yes.
So dorsoventral also sama dengan anterior posterior untuk human.
Okay.
Betul tak?
Ya.
Kalau kita kata kepala ke bawah, atas ke bawah, sama tak?
What is it? Can you repeat the question again, doctor?
Dari kepala ke kaki, dari atas ke bawah, refer to benda yang sama tak untuk manusia?
Ya. Itu benda yang sama, yes.
So dia itulah ayat dia. Maksud ayat tu.
Synonymous
kan?
Ya, synonymous. Okay, faham.
Faham kan?
Ya, faham saya. Alright.
Okay.
Ada lagi soalan?
Ada lagi soalan? No more? Okay. Siapa yang belum ambil attendance lagi? Boleh ambil. I'll share the QR code here. Make sure you scan ya. Ada yang absent tak? Saya check sekejap lah. Aiman ada tak? Aiman Hakim? Nur Hafiza Maisara?
Ada doktor.
Kenapa? You tak boleh scan kah?
Ya.
Okay. Okay, done.
Thank you doktor.
Aiman? Mana Aiman? Aiman tak hadir kan? Okay, tak apa. Dia absent lah. Kita kira dia absent. Siapa kena dia, suruh dia masuk ya. Suruh dia kena attend ya. Kita tak nak banyak alasan daripada pelajar. Because I don't really like to take any excuses from my students. If you have no question, I think that's all for today. Alright? And I'll see you next week. Kita ada satu assignment ya, sekejap lagi. Okay, this is the assignment. Can you see that?